Good afternoon, everyone. Thank you for having me. I'm very excited to be here virtually with you at Physiology and Pharmacology Conference. I'm delighted to present our study on mechanobiology of fibrosis. This has been a work that I've started during my postdoc at Washington University in St. Louis, and it's still an ongoing project now that I am a physician resident in pathology department at St. Louis University. Hope we can get the conversation going here and find some common ground. Some of the results I'm presenting here hasn't been published yet, so I would really appreciate your feedback. Let me start with what inspired me to work on mechanobiology of cells and tissues. I'm interested in cell matrix interaction in 3D collagen matrices and how these interactions on one hand can lead to healing wounds, but on the other hand can form fibrosis, and also it can play a very important role in tumor progression. Fibrosis is a serious health problem, and it's characterized by excess accumulation of extracellular matrix that can occur all over the body, as you can see in this slide, and can actually progress to organ malfunction. Fibrotic diseases include a wide spectrum of clinical entities of unknown etiology, for example, idiopathic pulmonary fibrosis. We call them idiopathic because we don't fully understand how it occurs or what are the processes that accelerate it or can slow it down. And there is still no definitive treatment available for it. Why is that? Because it's not a disease of pure chemistry. There's a lot of physics going on. Let me show you a video I took tracking a fibroblast for about 2 days in 3D collagen matrix. Look how these cells interact and remove and the matrix around it. And this highly dynamic interaction is driven by physical forces generated by a unique cytoskeleton system within the cell. And it's absolutely essential to a variety of biological processes such as morphogenesis, wound healing, cancer metastasis, and immune response. So these physical forces are important to study to understand the question of how cells can change the architecture and function of tissue and organ just by their interaction with the extracellular matrix. The question of how cells feel, how they remember, and the one that I consider the most important is how they can adapt the collagen and their microenvironment, which is called ECM remodeling. So I'm giving you a more visible example of fibrosis, which is Dupuytren's contracture. It's a hand deformity caused by excessive pulmonary fibrosis that can lead to finger contracture, as you can see it right here, and can cause by contractile myofibroblast under the skin. These are well-differentiated cells, essential for wound healing, that are responsible for tissue remodeling. But this is what happens when they get hyperactivated, where they shouldn't have. And you can see here that quiescent myofibroblast, actually fibroblast, in normal hand can turn into hyperactivated myofibroblast, aligning the matrix around them and forming these fibrotic traction fibers. So our focus is to look into this remodeling and this transition, and to be able to stop this, we are asking these questions. What are the mechanisms that cells use to interact with the extracellular matrix? What are the mechanisms of fibroblast to myofibroblast transformation, and can we stop this? So this is an outline of my journey. So we started with some fundamental questions on what the mechanisms is behind the fibrosis. To answer this question, we first started to look at fibrosis at tissue levels, then at individual cellular level, and then even smaller at subcellular levels. And these basic science knowledge at different levels actually helped us to come up with a few ideas on how we can stop and possibly prevent the formation of fibrotic tissues. And now, and our ongoing project is how we are planning to use this knowledge in clinic. So first step, we started with a simple model to study the cell matrix interaction. This is a picture of our cells in a 3D collagen matrix. I made these tissue constructs with human dermal fibroblast immersed in rat tail collagen type 1, which is in green here, and cells are in yellow. And I did the live cell imaging and followed each single cell from the very beginning under confocal microscope as they remodel the extracellular matrix around them over time. And then we analyzed the volumetric imaging stacks, as you can see right here, for each individual cell. So what do we know about the remodeling, and what do we not know? We know that remodeling extends over many cell lengths and can shrink the tissue down by communicating over very long distances. But what is not known, however, is what happens locally in the close proximity of each cell, how cells start interacting with the collagen. So first asked how isolated human dermal fibroblasts adapt their morphology and structure and their microenvironment in response to being embedded in a collagen gel from the very beginning. As expected, cells were initially round and they periodically extend and retract very small processes, and as these extensions and retractions progress, the collagen would be drawn in and accumulate in the vicinity of the cell. So we developed a computer code to measure changes in collagen density around the cell over time, and this graph is actually what we observed at level of cell processes. We estimated the density of collagen around the cell over time at several distances away from the cell, and as you can see, there is a significant increase in collagen concentration as a function of time, and actually it somehow can reach a steady state over time scales of hundreds of minutes, and they kind of stabilize. And at each time point you can see most of the collagen accumulation is around the immediate vicinity of the cell, and it decreases as you go further from the cell. And these different colors represent different distances away from the cell, and again the big change in the beginning shows the importance of the first several hours of cell ECM interaction that we mainly focused on that. So then we looked even closer at each cell, and we noticed the significant portion of this accumulation happens around these little protrusions, which indicates that protrusions plays a key role in accumulating collagen at early stages. Here you can see an SHG signal of cells and collagen separately, and the combined image here, and as you can see, just even very small protrusions can accumulate more collagen than the cell body. Then we asked, what are the main cellular components responsible for the formation of the cellular protrusions? And to answer this question, we disrupt different components of cytoskeleton, including microtubules and actin filaments. As you can see, this is a control cell, and this is when we disrupt microtubules with nacatazole in our tissue constructs, and this is where we disrupt actin filaments by treatment of the tissue with cytoclasine D. Is a 3D construct of a tissue with cells treated with nacatazole. So their microtubules are depolymerized, and we found that with depolymerization of microtubules, cells lose their processes and they become round, as you can see when you compare them with control condition here. However, they're still contractile and they can remodel the matrix a little bit around them, as you can see here, but obviously not as efficient as when they develop protrusions like this. So lack of protrusions due to disruption of core microtubules leads to a reduction in tension at the cell matrix interface. So this shows the importance of cell protrusions in accumulating collagen and generating tension. Next, we pre-treated fibroblasts with cytoclasine D, which is an inhibitor of actin polymerization, to study how cell matrix interaction is affected by disruption of actomycin contractility. And as expected, no collagen accumulation was observed around the cytoclasine D treated fibroblast. On the other hand, cell protrusions, as you can see them here, grew in both length and number, which is very interesting. This indicates that formation of protrusion is regulated by a mechanical force balance between actomycin contractility and microtubule extensions. Without microtubules, as you can see here, there is no protrusions. But as tension in the cytoskeleton decreases by disruption of actin, microtubules experience lower compressive forces and can grow in length and number. Example of how lowering the actomycin contractility with different drugs like cytoclasine D and Y27632 treatment, which both decrease the contractility of the cell, can lead to lengthening of the protrusion by unrestricted microtubule polymerization. The green in both is microtubule, in red is actin. Here's a schematic summarizing these findings. First, cell protrusions played a key role in accumulating collagen, and these protrusions contain both microtubules and actin filaments, and actin filaments experiencing these contractile forces and therefore generate this compression on top of the microtubule, and these compressive forces can in turn restrict the excessive growth of microtubules. So we conclude that there is a balance between actin contractile force and microtubule growth. So if it disrupts these compressive forces by depolymerizing actin, then microtubules are experiencing less compression and they can grow excessively in both length and number. Cells that are unable to form protrusions, the nacatazole pretreated cells, accumulating significantly less collagen around themselves and less remodeling. So now that we know how important those are in the process of remodeling and occurrence of fibrosis, we were wondering, can microtubule disruption stop progression of fibrosis? Well, it's not that simple. As you can see here, disruption of microtubules before embedding the cell within the matrix significantly reduce collagen alignment compared to control because cells lose their protrusions. if you compare these two, you can see significant collagen alignment here versus the collagen, the cortisol treated cells. But if you wait until the cells are already adapted to their environment and then disrupt the microtubules, it results in retraction of that protrusion that has been formed and result in higher collagen alignment. So the main message here is we found that fibroblasts use completely different mechanisms in early, which I think the most treatable stage of these interactions, and that their responses to drug can be the opposite of what they would be in the later stage. So by now we hypothesize that there is this two-way feedback between protrusion and collagen that help the tissue become fibrotic. So on one hand, these cell protrusions interact with the matrix and remodel and align the collagen fibers in their vicinity, and on the other hand, this aligned fibers also stabilize this protrusion and help them to promote the growth of the protrusion in the direction of collagen alignment. So we think that there is this two-way feedback between collagen alignment and protrusion stabilization that enables fibroblasts to become spread, polarized, and contractile over time. So at the end, they would be able to form these traction fibers in between to form fibrosis. Then we ask how the protrusions does this, what is the mechanism, and we've measured the strain generated around the cell protrusions using this strain mapping technique. And as you can see, there's this spectrum. As you go toward the red, it's more tension, and blue is compression. And what we expected to see first was when you put in a cell in a uniform collagen matrix, the cell would draw collagen in at the end using processes like this, and after a while you got like concentration of collagen around the cell. So we expected to see most of the tension here at the tip, but what we saw, as you can see, is totally different. So you can see that there is compression at the tip and a gradient of tension laterally to the cell process. And then when we went back and checked the actual microscopic images, these two are 3D constructed construction of that same cell at two different time points that we mapped for strain. We realized that at this stage, this protrusion is growing between these two time points. So it compressed the matrix while growing, and at the same time pulling on the collagen and generating tension laterally to the protrusion. So this is another example in this picture of how collagen would draw in and would accumulate laterally to the cell processes, this is exactly where the cells exert the most tension. And if you look closer, growing tip is actually obvious in this image. So from the tip they grow, and from sides they attach to the collagen and pull on them. Then why cells using this mechanism? And first of all, we know that cells need to be under tension to function normally, so they pull on their surrounding matrices to generate the tension they need. But why they're doing this by their side compared to just only with their tips? And the mathematical model here gave us the answer, which is much more efficient to pull on the matrix by their side, as it requires to exert much less energy to generate their required tension. Next we ask how much force is generated when the protrusions is retracted, and having the strain field from the experiments and using the theoretical collagen fibrous model, we kind of calculate the amount of force generated by a protrusion, which was in the order of like tenth of nanonewton. So we saw our strain mapping analysis also validate the two-way feedback hypothesis. So the protrusions can pull on the collagen fibers laterally and align them, and in turn the aligned fibers also stabilize the protrusion and help it to grow more. So cells talk to the environment and it talks back. So the cycle of protrusion, retraction, and growth continues to transform this cell from one that has projections all around it isotropically to a contractile polarized cell like this that can actually form distraction fibers and communicate with the neighboring cells as well. So we think cells does all these to get stabilized and polarized and subsequently develop this the required tension efficiently. So we think this recursive feedback between cells and ECM not only helps the cells gain polarity, but also helps the transition from fibroblasts to myofibroblasts. So to show that we grew our tissue in a cruciform mold like this, and in the middle of the cruciform specimen, like this, there is isotropic tension, and then as you move further out, the stress field switches out from isotropic to an isotropic tension down the road, right? So cells here, like this, are experiencing isotropic tension versus here that are expressing a directional tension and an isotropic tension. And the data look like this. Red here represent fibroblast cells stained for F-actin, which shows the level of contractility, and the results clearly showed that cells under tension and isotropy develop much more tension compared to the cells in the middle of the specimen where they experiencing isotropic tension. So here is also a theoretical model based on our hypothesis, which closely agrees with the experimental results, indicating that exposing cells to a uniform stress field can prevent hyperactivity and hypercontractility of fibroblasts. So now I'm back to my initial questions, and so we realized protrusions are the key to hyperactivation of fibroblasts, and these protrusions engage in a two-way feedback with the matrix to polarize the cells, and a disruption of these the two-way feedback either by inhibition of protrusions or generating isotropic stress fields can inhibit hyperactivation of fibroblasts. And also, we found out microtubules are the key components of these protrusions, right? So we aim to target microtubules at early stages. But how can we do this? We already talked about using chemicals, and then we learned how important tension and isotropy is. So we thought about mechanical treatments, for example, exposing tissue to tension in different directions to affect cell behavior mechanically. This could be a very great therapy, but it's actually limited to the location and accessibility of the tissue, right? So our next step, which is we're very excited about and we're still working on it, is to study the effect of electric field on progression of fibrosis. At the end, I want to thank my mentors, our collaborators, our group members, funding, and Dr. Khoury for the invitation. Thank you. And hope the internet connection would cooperate and we can chat. But if not, I am providing you with my email. I would appreciate your feedbacks or comments, or I can take your questions. Um, Thank you very much again, and have a great rest of the day. Thank you.